Hi dear students, today we are going to talk about two eminent scientists who played a very good role in the history of molecular biology. They mainly include Salvador Luria and Max Delbruk. Salvador Luria, he was an Italian doctor and he was quite bored about medicine during the 1940s, and Delbruk was in turn a physicist, and he was also a German who was totally bored of physics, in fact. And due to the situation of the World War, the fund shortages which was being caused by the World War, as well as the inconveniences they had to conduct their experiments in their own countries, these two scientists migrated towards the United States at two different times, and they happened to meet over there, which led to a very big collaboration which developed a new branch in molecular biology based on the bacteriophages, that is, the viruses which eat bacteria. And these bacteriophages were further used as even genetic tools in molecular biology. So let's go today to explore these two scientists, and their experiments have even led them to the Nobel Prize. So let's go into their experiment. Known for their names, this experiment is referred to as the Luria and Delbruk experiment. So let's go into the details of it. Luria and Delbruk in 1943, they presented the results of their experiment as a paper in genetics titled Mutations of Bacteria from Virus Sensitivity to Virus Resistance. And this experiment was a major landmark in the history of genetics because it was a good evidence against the Lamarckism, and it also proved that mutations were as a result of random and spontaneous events which would happen in the case of bacteria. Now this is Max Delbruk and Salvador Luria, courtesy to the Cold Spring Harbor Laboratory in 1953. And this is a picture which when they were awarded the Nobel Prize in Physiology or Medicine in 1969, which was also shared by Alfred D. Hershey for their work which was being done in the field of bacteriophages. Now, I have mentioned this earlier about Delbruk, and before going into the experiment, I would like to draw your attention into something which we already know right now. When you take a bacteria, okay, and plate it onto an agar plate. This is a agar plate, and if you take the bacteria and into that you add a bacteriophage containing sample and shake it and put it onto the agar plate, you can see that after solidifying and after incubation, it has been found that at the regions where the bacteria is being eaten up by the virus, there will be some clear zones, and these are referred to as the plaques. So the regions where the viruses or the bacteriophages have eaten up the bacteria, you can see some cleared areas, and that is referred to as the plaques. So the evidence of the growth of bacteriophages on the plate can be seen by the presence of certain bacteria which are being found to be sensitive to the presence of these bacteriophages. So, and at the same time, you can see certain areas here where the bacteria is not killed by the bacteriophage. Such areas are referred to as the resistant bacteria. So, in the coming plate, or in the given plate which I have shown you here, you are having two different types of bacteria. One is the bacteria which was resistant to the phage, and they have forming colonies. Those are the cream color ones which you see here. But at the same time, there are regions where there is cleared zones or the cleared plates are being formed, representing the regions where the bacteriophage has eaten up the E. coli in the particular plate. So keep this in your mind, and do remember that we have two different types of colonies on this particular plate. So we'll be continuing in the next session the details of this experiment. This is the knowledge that we already have right now, but at this time of Max Delbruk's experiments time, they did not have this information in their mind. So let's continue in the next session.